Welcome to Word of Hope, Ovarian Cancer Podcast, brought to you in partnership with Word and Libby's Hope. For more links and information on this episode, visit wordofhopepodcast.com.
Welcome to Word of Hope, Ovarian Cancer Podcast, Episode 8. I'm Nate Manahan of Women's Oncology Research and Dialogue, also called Word, and I'm joined again today by...
And I'm Paul Cacciatore. I'm the founder of Libby's Hope, which is an educational ovarian cancer website.
We're glad to be back. We did the first 7 episodes kind of all together. It was a couple days of recording. But now we're here at episode 8, and we're looking at something that I think is really important. And you did an incredible job at a post, I guess it was a little over a week and a half ago.
Yeah,
and
that's right.
This post was by far a very exhaustive look at this study. And when I printed it out to be able to read it, it was 65 pages. And I would guess, Paul, this is probably one of your most exhaustive posts. I think it probably is maybe your longest post, I would think, by far.
Oh, absolutely, Nate.
Yeah. And so basically this was a summary of a very large research project that we'll call TCGA for short. Paul, could you give a description and a basic understanding what TCGA is?
Sure. TCGA, it stands for the Cancer Genome Atlas. It was started in 2006. And what it is it was a pilot program put together to look at genomic and epigenomic changes. We'll get to that a little bit later. In 3 different types of cancer. Glioblastoma, a form of brain cancer, aggressive form of brain cancer, ovarian cancer, and lung cancer. And the thought was they would do those first 3. And then after that, assuming everything goes well, they would look into the feasibility of basically mapping out or looking for changes in all major types of cancer. And so that's kind of where the idea got started. It's a joint collaboration between the U.S. National Cancer Institute and the National Human Genome Research Institute, both of which are institutes within the National Institutes of Health. The bureaucracy kind of gets big, but those are 2 of the institutes within the 20-some institutes and agencies within NIH. And so they worked together on this. And what they did is they took 489 high-grade serous epithelial ovarian cancer tumors and basically went ahead and analyzed them. So that's, in a nutshell, that's what they were intended to do. What they wanted to do was look at several types of genomic changes, and we could talk about those more in a bit. They fall into various categories that they looked at. And what they want to do is compare it basically to tissue taken from those folks who contributed the tumor samples and compare it to their normal cells. So whenever you have cancer, obviously adjacent to it, very closely, you can have normal cells. And so the theory is if you can lay one on top of the other, you can look at the genomes of both and see where the differences lie. And by doing that, hopefully it'll give you some lead in on the nature of cancer, what drives it, how it survives, how it proliferates, things along those lines.
We said this was about high-grade serous ovarian cancer. Paul, can you describe a little more what high-grade serous ovarian cancer is?
Sure. Let's just start with that. Let's just break it up, into its terms. High grade, or the word grade, tumor grade is really the term that we're talking about. I think the easiest way, Nate, to understand tumor grade is it's a system. It's a rating of generally 1 to 4. There are different systems for different types of cancer, but as a general matter, what they're looking to see is how far away, when you look under a microscope, characteristically, how far away has the cancer cell gotten away from a normal cell? So if you think about it, as adults, we have what are known as differentiated cells. All of our cells have a function. We have skin cells, we have heart cells, lung cells, things along those lines. They all have a function, and they do it pretty well, and they stay the same throughout our lifetime, at least our adult life. And so when you look at grading, the thing with cancer cells is they are able to kind of get back or move towards our cells, back at our earlier development, embryonic development, childhood development. Because what happened is cells were differentiating as we went from a 2-cell organism into like a trillion-cell adult. And back when we first started, developing as embryos, it was important for cells to be kind of uniform in the sense that they could almost become anything. And then the epigenetics, the genomics allow it to basically transform into what it needs to be. So really the grading system is nothing more than comparing how close is this cancer cell to a normal cell. the more it gets away, the more it becomes undifferentiated compared to our existing cells, the higher the score. And usually what happens is you'll hear some people, a lot of these terms are used, kind of thrown around loosely. You'll hear, for instance, a grade 4 tumor is an aggressive tumor. Well, there's a lot of technicality in that. I think really the best way to understand a grade 4 or the higher grades is that the cancer has moved away from the characteristics of a normal cell into a cancer cell. And we know the characteristics of cancer cells. They're very fast, quickly proliferating cells, unlike our normal cells. On serous ovarian cancer, just real quick, it's just one of the subtypes of epithelial ovarian cancer, and it is the major subtype, more common, attributable for more ovarian cancer deaths than any of the others. And so it's arguably the most common epithelial ovarian cancer to take a look at.
Paul, what are the real statistics and demographics of this study? And does it confirm what other studies have found of high-grade serous ovarian cancer, such as progression-free survival and overall survivorship?